Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on pancreatic juice. Now, first of all, the composition of pancreatic juice, what the pancreatic juice is made up of. Now, the color. So, as far as the color of pancreatic juice is concerned, so pancreatic juice is the transparent, colorless fluid, and it is isotonic with the plasma. Daily in our body, 1200 to 1500 ml of pancreatic juice is secreted, and the specific gravity of the pancreatic juice is 1.0102 to 1.018. Pancreatic juice is slightly basic, it is slightly alkaline, and the pH of the pancreatic juice is 7.8 to 8.4. So this is the range of the pH for the pancreatic juice. And in this pancreatic juice, high concentration of bicarbonate ion, that is HCO3 minus, is present, and this concentration is 4 to 5 times greater than that of the plasma's bicarbonate ion concentration. Now, the composition of pancreatic juice, the components of the pancreatic juice. So in the pancreatic juice, 99.5% water is there and 0.5% solids are present. Now in the we can divide these components of the pancreatic juice into organic and the inorganic substances. Now in organic substance, mainly the enzymes that are present. So the enzymes which are present in the pancreatic juice are the amylase, lipase, protease, trypsin inhibitor, and few traces of albumin and the globulin are also present. Now in inorganic substances, so mainly the positive ion, cation, Na plus, then the K plus, then Ca twice plus, Mg twice plus, Zn twice plus, HCO3 minus, these are the anion, negative ion, Cl minus, SO4 twice minus, and HPO4 twice minus. So these are the anions that are present. Now the main discussion, the main topic, or the important part for this topic, that is the pancreatic juice, is the discussion of the enzyme, which are the enzymes, in which form they are secreted, how they are activated, and how they are involved in the process of digestion, this pancreatic enzyme. Now the pancreatic acini. So in our previous lecture on the histology of the pancreas, we have learned that the pancreatic acini, they secrete the pancreatic juice, and in the pancreatic juice, four main type of enzymes are present. So these enzymes are, first, the first enzyme that is present in the pancreatic juice is the pancreatic alpha amylase, and the action of pancreatic alpha amylase is similar to salivary amylase. So we have studied the process of digestion in the mouth. So in that we have learned that the salivary amylase is act upon the carbohydrate. Similar way, the pancreatic alpha amylase, which is present in the pancreatic juice, also act upon the carbohydrates in our food. And basically pancreatic alpha amylase is hydrolyzing the glycogen and starch, and this amylase convert glycogen and starch into the disaccharide. Now here in the figure, this is the structure of the starch is shown, and we know starch is made up of many glucose molecule, and the arrangement of glucose molecule is in the two form, that is the amylose and amylopectin. And here you can see this, that starch is acted upon the pancreatic alpha amylase. And what this alpha amylase is doing, so it is digesting starch and converting starch, it is hydrolyzing the starch, and there is a formation of maltose, then trisaccharide, there are the larger oligosaccharide, that means the limit dextran are formed, and in the limit dextran there are 4 to 9 glycosyl units, that means the glucose molecules are there. Okay, and the linkage is alpha 1 6 glycosidic bond is there between the glucose molecules. Then when the starch is digested, alpha dextrine is formed. Alpha dextrine is the oligosaccharide, and once again the bond is alpha 1 6. Then isomaltose is formed. So we can say that the pancreatic amylase hydrolyzed starch, and then there is a formation of maltose, then trisaccharide, that is the limit dextran, then alpha dextrine, isomaltose. All these are formed due to the action of the pancreatic amylase. So this was the first enzyme. Now moving on the second enzyme. In the pancreatic juice, for the digestion of lipid, lipase are present. So pancreatic lipases are also known as the lipolytic enzyme, and the example for the lipolytic enzymes include pancreatic lipase, cholesterol ester hydrolase, and phospholipase A2. Now, pancreatic lipase is a powerful lipolytic enzyme. That means pancreatic lipase is the strong lipid or the fat digesting enzyme. And the action of the pancreatic lipase that it hydrolyzes the natural fat. That means the triglyceride, which are present in our food in the form of fat or oil. Now, this pancreatic lipase hydrolyzes the triglyceride and converts the triglyceride into glycerol, ester, and fatty acid. Now, here in the figure, this is the structure of triglyceride on the left hand side, and we know in the structure of triglyceride, this is one glycerol molecule, and it is attached with three fatty acid. So these are the three fatty acid molecule, and this bond is the ester bond. Now here on the arrow you can see the enzyme lipase is shown and H2O. So what we are doing, that we are adding this water molecule into triglyceride, and we are hydrolyzing this triglyceride with the help of lipase. So what happened, one fatty acid will be removed. Let's say this this fatty acid is removed, so triglyceride is converted into diglyceride. Now this diglyceride are further hydrolyzed by lipase, so we will add one water molecule and then removal of one fatty acid. Okay, so now we are having the monoglyceride. So in monoglyceride you can see this, there is a one glycerol molecule attached with one fatty acid. Now if we further this monoglyceride, if it is further acted upon by lipase, so this last fatty acid will be also removed, and this bond, so this bond is the ester bond, so this bond will be broken down, and ultimately we will be having the glycerol and the fatty acid. So you go through this figure, and with this figure you will get the idea how the triglyceride is hydrolyzed by the lipase enzyme and how the monoglyceride or the fatty acids are formed. So this was the theory point, and this is the figure. Now another enzyme is there, and the enzyme name is the cholesterol ester hydrolase. Now here, this is the structure, of cholesterol ester. Now in the structure of cholesterol ester, we will remember, so this unit, this one is the cholesterol, and this one is the fatty acid. Now the name of the enzyme is the cholesterol ester hydrolase. And what this cholesterol ester hydrolase enzyme is doing, so it is breaking this bond. So this bond is between the carboxyl group and the hydroxyl group, carboxyl group of fatty acid and hydroxyl group of cholesterol. So this bond is broken between carboxyl group of fatty acid and the hydroxyl group of cholesterol. So due to that, due to the splitting of that bond, what happens, there is a formation of cholesterol and there is a removal of fatty acid. So we can say that cholesterol ester hydrolase enzyme act upon cholesterol ester, and it converts cholesterol ester into cholesterol and fatty acid. So now we are moving forward. Then this enzyme phospholipase A2. Now this phospholipase A2 is secreted in the inactive form, and this inactive form is known as prophospholipase A2. Now the enzyme name trypsin. Now this trypsin act upon inactive prophospholipase A2, and due to the effect of trypsin, this prophospholipase A2 is converted into active phospholipase A2. Now this active phospholipase A2 act on lysolecithin and lysocephalin, and this phospholipase A2 converts lysolecithin and lysocephalin into phosphorylcholine. So this is the function of phospholipase A2, how it is activated and what is the role it is playing in the digestion. Now the digestion of protein. So for the protein digestion, there are pancreatic protease enzymes are present in pancreatic juice, or they are also known as the proteolytic enzyme. And the example of proteolytic enzyme include three endopeptidases, and this endopeptidases example are trypsin, chymotrypsin, elastase. And in the pancreatic juice, two exopeptidase enzyme, carboxypeptidase A and carboxypeptidase B. So first we will discuss about the trypsin, chymotrypsin and elastase, and then we will study the effect of carboxypeptidase A and B. Now first, trypsin. Now the trypsin is the most powerful proteolytic enzyme. That means it is a very strong protein digesting enzyme present in the pancreatic juice. Now from where this trypsin is secreted and in which form it is secreted? So first of all, the trypsin is secreted in the inactive form, and this inactive form of trypsin is known as trypsinogen. And this inactive trypsinogen is activated by the enzyme enterokinase. The other name for the enterokinase is the enteropeptidase. So, enterokinase act upon trypsinogen, and this trypsinogen is converted into active trypsin. Now, this enterokinase enzyme is secreted by duodenal mucosa. So here in this figure, as you can see, stomach is followed by duodenum. So this is the wall of duodenum, internal wall, that is the duodenal mucosa. And in the duodenal mucosa there are epithelial cells are present around the lumen, and this epithelial cell, they secrete the enzyme enteropeptidase, and the other name for the enteropeptidase is the enterokinase. Now what this enterokinase is doing, so as you can see, this enterokinase act upon inactive trypsinogen, and due to the effect of enterokinase enzyme, this inactive trypsinogen is converted into trypsin. Now what this trypsin is doing, so this trypsin will act upon the remaining trypsinogen, and the remaining trypsinogen will also be converted into active trypsin. Then trypsin also act upon chymotrypsinogen. So due to the effect of trypsin, chymotrypsinogen is converted into chymotrypsin. Then we are having procarboxypeptidase. This is in the inactive form. So this procarboxypeptidase is acted upon by trypsin. So due to the effect of trypsin, procarboxypeptidase is converted into active carboxypeptidase. And for the activation, this enterokinase is also there. So this enterokinase is also participating, and this enterokinase also responsible for the activation of procarboxypeptidase into active carboxypeptidase. So these are the conversion you need to remember, which is the enzyme secreted in the inactive form and who is activating enzyme. Now here, so trypsin, now what the role that active trypsin is playing. So the active trypsin hydrolyzing the protein, and it is digesting the protein into proteoses and the polypeptides. Then, as we have discussed, that trypsin activates the remaining trypsinogen and convert it into active trypsin. Okay, then chymotrypsinogen. Chymotrypsinogen in active form, so it is acted by trypsin and converted into chymotrypsin. Chymotrypsin function hydrolysis protein into small polypeptide. Then another enzyme, that is elastase. Elastase is secreted as proelastase, and this proelastase is also activated by trypsin, and proelastase is converted into elastase. Elastase digest elastin. Now here, carboxypeptidase. So first of all, there are two carboxypeptidase in the pancreatic juice, carboxypeptidase A and carboxypeptidase B. And both these carboxypeptidase, they are secreted in the inactive form of pro carboxypeptidase A and pro carboxypeptidase B, and they are activated by enterokinase and trypsin. So enterokinase and trypsin, they act upon inactive procarboxypeptidase A and B, and this procarboxypeptidase A and B, they are converted into active, carboxypeptidase A and B by the trypsin and enterokinase enzyme. Now what this carboxypeptidase are doing? So here you see, this one is the polypeptide is shown. Now polypeptide, we know polypeptide means many amino acids joined together by CONH peptide bond. Okay. Now what this carboxypeptidase A is doing? So it cleaves the carboxyl terminal amino acid. It is removing, it is breaking the bond, peptide bond, and it is removing amino acid. And this amino acid which is removed is having aromatic or the branch aliphatic side chain. Let's say over here, this is the polypeptide is shown, this one, this is the structure of polypeptide, and this carboxypeptidase A, is breaking this bond. So this bond is broken down. So this amino acid molecule is released, and this released amino acid molecule is shown over here. Okay, so this is the released amino acid, and in this amino acid, this functional group RDAS, so RDAS is aromatic or it is the branch aliphatic side chain, and the name of the amino acid RDAS, all the amino acid except arginine, proline, lysine and hydroxyproline. And generally the aromatic or the branch aliphatic side chain amino acid are preferred for the action of carboxypeptidase A enzyme. So in short, this carboxypeptidase A enzyme is acting upon the polypeptide, and it is removing terminal amino acid, and this terminal amino acid which is removed is having the aromatic or the aliphatic branch side chain. That is the meaning, and the name, all the amino acid, terminal amino acid can be all the amino acid, but except this amino acid are not. So these are not involved: arginine, proline, lysine, hydroxyproline. Okay. Now carboxypeptidase B. Now again, this enzyme also acting upon polypeptide. It is also cleaving bond, peptide bond, and it is removing carboxyl terminal amino acid. So let's say this amino acid is removed, it is released, and it is shown over here, and the released amino acid is having RDAS. So what is RDAS? So it is a basic side chain. So the amino acid which is removed, which is released, they are arginine, lysine and ornithine. And here you can see on left hand side polypeptide N residue, that means N amino acid were present, and now this polypeptide N minus 1. So one amino acid is cleaved off, one amino acid is removed. So this is the action of carboxypeptidase A and B. Now nucleases. So in the pancreatic juice, ribonuclease is present. So this is involved in the digestion of RNA. Deoxyribonuclease is present. This is involved in the digestion of DNA. Now where this nucleases, so this nucleases will act upon phosphodiester bond. Okay, so this nucleases, they are breaking the phosphodiester bond of RNA and DNA. So once the phosphodiester bond are broken down, what happens? The nucleotides are released. So from RNA, ribonucleotide will be released, and from DNA, deoxyribonucleotides will be released. So nucleases, then enzyme collagenase. This enzyme is also activated by trypsin, and this active collagenase digests collagen. Now, interesting point we need to notice regarding the pancreatic juice, that the acini of the pancreas, the glandular epithelium, the pyramidal shape cell, they secrete pancreatic juice. In the histology we learn, and in the pancreatic juice, this trypsin inhibitory factor is also secreted by this acinar cell. Same time, when the trypsin was released by this cell, so this cell, the acinar cell, they are secreting the trypsin. At the same time, they are also producing this trypsin inhibitor. Now the reason is that trypsin is a so strong proteolytic enzyme that it can digest the entire pancreas. So to protect the effect of trypsin, this trypsin inhibitor act on this trypsin, and it is inactivating the trypsin, so that the trypsin cannot digest the pancreas. So this point we need to remember, trypsin inhibitor and what is the role of trypsin inhibitor. Then finally, the function of the pancreatic juice. So the pancreatic juice is the major source of digestive enzyme, and in our food, proteins, carbohydrate, fat and nucleic acid are present. So digestion of protein, carbohydrate, fat and nucleic acid is possible due to the enzyme which are present in the pancreatic juice. Then neutralizing effect of pancreatic juice. See, in the chyme after the digestion, what happens? The chyme leaves stomach and enters into the duodenum, and in this chyme HCl is present. Now we know the HCl is the acid. So to neutralize the effect of this acid, this bicarbonate ion which are present excessively in the pancreatic juice, and this bicarbonate ion, they are neutralizing the effect of acid HCl. So these are with this we have completed this presentation lecture. So here in this lecture, the main thing is the name of the enzyme, their inactive form, how they are activated, and what is the course of their action, how these enzymes are involved in the digestion process. This thing we need to remember. So with this we have completed this presentation lecture. on the pancreatic juice. I hope you have enjoyed this presentation lecture, and I also hope that this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Kushti sir. I am from Ahmedabad, India. Bye bye. Namaste.